Hello everyone, this is MH Mubashir again and welcome to the BTC 509 Bioinformatics tutorial. And in this tutorial I will give you some basic idea about the eukaryotic gene and prokaryotic gene and their structures. Okay, so this is all about theory. So this tutorial I won't be showing you anything practical. This tutorial will be all about some theoretical knowledge about prokaryotic gene structure and eukaryotic gene structure. Okay, let's talk about this picture. Okay, this is the basic structure of an eukaryotic gene, and we all know that from DNA we get mRNA and from mRNA we get protein, right? But in case of eukaryotes, there is a term known as pre-mRNA, because this mRNA or pre-mRNA need to be modified to become a mature mRNA. So this mature mRNA is present in the cytosol or cytoplasm, and this pre-mRNA is present in the nucleus. And you can see a basic difference between pre-mRNA and mRNA is that you can see some introns here in the pre-mRNA. That means this is not matured yet, and this is the matured mRNA, the red portion, and these are the exon portion of the RNA or the coding portion, the coding region of the mRNA. Well, from this region, basically from this entire red region, we will get a protein. Well, so there are some blue structures here we can see, and you see this is known as 5' untranslated region. So this part won't be translated, and this is another part, 3' untranslated region. So this is 5' untranslated region, and this is 3' untranslated region, and within this we have open reading frame, which is known as ORF. We'll be doing some analysis for searching the UTR and ORF later. Okay, for now just remember we have 5' untranslated region, we have ORF, and we have 3' untranslated region. And from this untranslated region we get 5' cap, and from this 3' untranslated region we get poly-A tail. And we all know that this 5' capping and poly-A tail is very prominent and distinguishing feature of eukaryotic mRNA, and this is very important because this mRNA has to travel a lot of area, and for those traveling protection is very much necessary, and this 5' capping and 3' poly-A tail will give the protection to this mRNA. Okay, so now we are not having intron at the end, we are having only exons, and this means these exons will only be producing the protein or the amino acid sequence. Let's look into some other structures of this eukaryotic gene. So this part is known as regulatory sequence, and this is at the end, this is also known as regulatory sequence. And this is the upstream region, and this is the downstream region. At the upstream region we have enhancer or silencer, and at the downstream region we also have enhancer or silencer. What is the function of enhancer or silencer? So basically the function of enhancer is to speed up the process. Okay, and the same thing is happening for silencer. The function of silencer will be to stop the process of transcription and then translation. Okay, so this is the region where the promoter stays. It's at the upstream region, and we have two parts of the promoter. One is the core region, another one is the proximal region. The core region is most important for us, for our case, and we'll be also searching for promoter in our future tutorials. And let's look into the structure of promoter in a bit detail. Okay, so this is the structure of promoter basically. So we know that we have two parts of a promoter. One is the core region of the promoter, and another one is the proximal region. So in core region we have data box, and which is present in the -25 base pair upstream of the gene. That means if this is +1, that means the transcription will start. Then at the 25 base pair upstream we will have this data box. This is known as data box because we have the sequence TATA here, and here we have CAAT box as well. We have GC box, and these are some regulatory sequence which will regulate the function of the promoter. But then again, this is not very much important for us. So we have two parts of a promoter, proximal region and then core promoter region. And then at the end of the core promoter region we have this +1, which is the start of transcription, and then we have, like I said to you, UTR or the untranslated area, and then we have exon and then intron. This intron will be cut down, and then we have exon 2 and then exon 3, and then we have untranslated region again. Okay, and this side is we will be having 5' that will generate 5' capping, and this part will generate 3' poly-A tail. Okay, so that's the basic structure of the eukaryotic gene. I believe you have understood it well. So I think that's it for the eukaryotic gene structure. Well, let's talk about the prokaryotic gene structure. In case of prokaryotic gene structure, you can see it from here that they don't have any intron. Okay, so we don't have any intron in case of prokaryotic structure. So all the sequences here will be transcripted and translated. One interesting fact in case of prokaryotic gene structure is, here we have one single promoter, but with this one single promoter we have multiple genes. For say this case 1, 2, 3, 4, and we will get 4 proteins from these 4 gene structures. Okay, that means in case of eukaryotes you will get one promoter for one particular gene, but in case of prokaryotes you can also have one single promoter for multiple genes. Okay, and from this part to this part is known as operon. So from this part to this part will be translated. We have promoter, operator here, and at the above there are some other regulatory genes as well. So this is the basic structure of a prokaryotic gene. So here in case of prokaryotic gene we have a start codon, and most common start codon is AUG, and most common stop codons are these 3. Okay, another interesting fact is, for AUG we will get a specific amino acid which is methionine, but for these 3 codons we won't get any amino acids. Okay, so this is the coding region, and you can easily see it from here that with the AUG we will have some other mRNA part at the upstream region. Okay, and also we will get some mRNA at the downstream regions after the stop codon as well. So what are those regions? Let's talk about those. So this is a prokaryotic mRNA structure, and in case of prokaryotic mRNA you can see that we have a start codon here, and before the start codon in the upstream side we have Schindel-Garner sequence. That means we have this sequence in mRNA, and definitely we have this complementary sequence of Schindel-Garner in the DNA of that particular bacteria. So in case of prokaryotes you will see that kind of sequences. Okay, so we will search for prokaryotic gene or eukaryotic gene, and for that case remember you have to go for, or the server which we are searching for the gene will definitely go search for the sequence which is complementary for Schindel-Garner. Okay, another important feature for this prokaryotic DNA structure or gene structure is that at the end of the sequence it has multiple A, that means poly A site, and this definitely indicates the terminal site or the termination region. And also before this poly A we have several GC sequences there. Okay, and definitely they will form hairpin loop, and we all have already learned this thing during our undergrad, so I don't want to go through detail of it. So that's it, the basics of prokaryotic gene structure and the basics of eukaryotic gene structure.